Today, we are pulling back the curtain on one of biology's greatest secrets, the hidden control system that runs everything in every living thing. Seriously, from a single bacterium to you and me, there's a master plan at work, and we're about to decode it. Here's a mind bender for you. Your body has something like 200 different types of cells. Neurons, skin, bone, you name it. And yet, every single one contains the exact same genetic blueprint. So, how does one instruction manual create so many completely different things? That's the big mystery we're going to solve today.
So, what's the secret sauce? Well, it turns out the answer is both incredibly simple and totally profound. It's not about the genes you have, it's about which ones you actually use. It all comes down to a genetic on-off switch. And the official name for this is gene regulation. You can think of it as the master control program for the library of life. It's how a cell decides, okay, I'm going to read this instruction, but ignore that one, making sure it only produces what it needs, exactly when it needs it. This is the fundamental engine that creates all of life's amazing diversity from the same set of rules. And the logic is surprisingly simple. Imagine your DNA is a giant cookbook with thousands of recipes. Gene regulation is the chef. And that chef isn't going to make a soup, a roast, and a dessert all at the same time, right? No. The chef only opens the book to the cake recipe when it's time for dessert. When a gene is on, its protein gets made. When it's off, nothing happens. It's that simple, and this one trick prevents total chaos inside the cell and saves a ton of energy.
Okay, so what's actually flipping these switches? Well, the job is done by a special class of proteins called transcription factors. You can literally think of them as the fingers that push the buttons on the DNA. You've got two main types. Activators are the on button. They bind to the DNA and kickstart the whole process. And repressors, well, you can probably guess, they do the exact opposite. They latch on and block the process, shutting the gene down.
All right, so that's the theory. But how does this all play out in the real world? To see this system in its purest, most elegant form, we're going to look at some of the simplest life out there, bacteria. These guys are the undisputed masters of genetic efficiency. Bacteria use this genius system called an operon. Let's go back to that cookbook. Instead of having separate recipes for flour, sugar, and eggs, an operon groups all the genes for one job, like all the cake-related genes, together under a single master switch. It's an incredibly smart and efficient way to coordinate everything. A classic example of this is the lac operon in E. coli. By default, when there's no lactose around, a repressor protein just sits on the DNA, blocking everything. The switch is off. But the moment lactose shows up, it acts like a key. It binds to that repressor, changes its shape, and makes it fall right off the DNA. And boom, the path is clear, and the cell immediately starts making the enzymes it needs to digest that lactose. It's a perfect, on-demand system. And the key here, the real genius of it, is efficiency. I mean, why would you bother activating 3 separate genes when you can just flip one master switch? By grouping genes this way, bacteria make sure they don't waste a single bit of energy making proteins they don't need. It's just a beautiful piece of biological engineering. Okay, so bacteria are super efficient. Got it. But what happens when you need to build something way more complex, like a person? Well, the same basic principles are still there, but we've added some incredible new layers of control. Let's start with a number. The human genome, our entire genetic instruction book, has roughly 30,000 genes, okay? Just lock that number in your head for a second. 30,000. But now get this. The number of different proteins our bodies can make is estimated to be over 400,000. So, wait a minute. How do you get more than 10 times the number of proteins from your genes? The math just doesn't seem to add up, right? The answer is a total game changer called alternative splicing. It's like having one core recipe that you can edit to make a bunch of different dishes. A cell can take the initial message from a gene and literally snip out different sections, stitching the remaining pieces together in new combinations. This lets just one gene create a whole family of related but different proteins. This is a huge reason we're so complex. And that's not all. In creatures like us, the control is multi-layered. First, the DNA itself can be physically packed away so tightly that it's just unreadable. Then you have epigenetics. Think of them as chemical sticky notes on the DNA that tell a gene to be active or quiet. And finally, many genes need a specific combination of several different factors to turn on. It's like a high-security vault that needs multiple keys turned at the exact same time. Okay, so let's zoom out. All this microscopic control isn't just for show. It's what allows entire organisms to respond, adapt, and ultimately survive in a world that is constantly changing.
Plants are a fantastic example of this. Think about it. They're stuck in one spot. They can't run from danger or move to find more sun. So they have to adapt right where they are, and they do it through some incredibly sensitive gene regulation. This is all orchestrated by hormones. Auxin, for example, helps a plant physically bend towards a light source. Ethylene is a gas that tells a seedling that's hit a rock to slow down, get stronger, and grow around it. And abscisic acid is like the emergency brake during a drought. It tells the leaves to slam their pores shut to save water. These are all visible, life-or-death actions driven entirely by genes turning on and off. In animals, this constant regulation hits its absolute peak in a state we call homeostasis. This is the active, moment-to-moment job of keeping your internal world, your temperature, your blood sugar, your salt balance, perfectly stable inside a very narrow range, no matter what's going on outside. And animals have two main strategies for this. There are regulators, like us, who work really hard and burn a lot of energy to keep our internal state constant. Then there are conformers, who just let their internal state change along with the environment. Both approaches have their pros and cons. Now, if you want to see a true master regulator in action, just look at a bony fish. In the salty ocean, it's constantly losing water. To survive, it has to drink tons of seawater and then use its gills to actively pump all the excess salt out. But put that same fish in freshwater, and the problem totally flips. Now it's gaining too much water. So it stops drinking, produces a huge amount of dilute urine, and uses its gills to pull in what little salt it can find. It's a dynamic, two-way regulatory machine. And this really brings us to the core idea. Life isn't a static thing perfectly balanced with its surroundings. No way. It's a constant, active process of self-management. Life is more like a fire than a stone. It is a dynamic state of perpetual regulation. From the simplest bacterial switch to the staggering complexity of the human brain, the story of life is the story of regulation. And now, as we're learning to not just read this genetic control panel, but even to edit it, it begs the question, what new capabilities, what new cures, and what new challenges are we about to unlock? What story will we choose to write next?